Hi, everyone, and thank you so much for joining our IP Mass Spec workshop. I'm really excited to have Dr. Simone Sidoli here today as he'll share some of his expert tips and best practices to help improve data quality and reproducibility of your IPMS experiments. But before we begin the workshop, I just want to give everyone some more information about Protein Tech and who we are as a company. Protein Tech is a leading provider of life science reagents. The company was founded in 2001, and at that time, it was primarily a supplier of antibodies and immunoassays. And because we've been in the game for over 20 years, we do have an expansive portfolio of antibodies that cover about two-thirds of the human proteome, as well as many different research areas. We also offer immunoassays such as ELISA kits and IHC kits, and we also offer cell separation kits and antibody labeling kits. The company then later expanded to include more product lines. So we now have our humankind branded cytokines and growth factors. And all our proteins are expressed in human HEC293 cells. So you can be confident that they have the native folding, post translational modifications, and glycosylation that you would expect to see in human proteins. And that ultimately increases their bioactivity. We also have our Chromatec branded nanobody based reagents, and I'll talk a little bit more about nanobodies after Dr. Sidoli's talk. But nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, making them ideal for applications like immunoprecipitation, super resolution microscopy, and protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics. And they manufacture antibodies that are conjugated to oligonucleotides to allow for protein detection alongside your single cell RNA sequencing. So, as a company that is driven by scientists, we're here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. For convenience and testing, most of our antibodies are also offered in 20 microliter trial sizes. We also know the importance of product validation, and we do offer open access validation data for all our customers. Whenever we are testing new products, we do validate with relevant sample types, appropriate experimental conditions, and using knockout and knockdown models whenever possible. Also, as antibody experts, we have in depth knowledge of immunoassay applications, and we offer outstanding technical support for our customers. We also stay on top with cutting edge technology, such as our proprietary antigen specific B cell cloning and engineering platform that we use to manufacture our recombinant antibodies. With 7 inventory sites and manufacturing and research sites on 3 continents, we are able to offer next day delivery to many areas in the world. So, as a company, we do measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. So, this graph here on the left shows that over our company history, our products are being featured in more and more publications each year. And as of now, we have over 310,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. I also quickly just want to highlight that as a company, we do strive to make the searchability of our products on our website as user friendly and informative as possible. We recently launched some new updates on our website to help our customers select the right antibody. This includes our unique 3D epitope viewer that allows you to interact with epitope structures and explore antibody binding sites so that you'll know exactly where our antibodies bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need, such as our Immunogen Region Selector toggle that allows you to filter for antibodies generated from your region of interest. Additionally, you no longer have to filter through all the variations of an antibody for the search results. You can view all formats of an antibody, including conjugations and buffer formulations, on just one page, which ultimately streamlines your process for antibody selection. So, with that, I would like to introduce our speaker for today and get things kicked off. Dr. Simone Sidoli is here with us. So, Dr. Sidoli approached Mass Spec as a trainee in the Department of Chemistry at the University of Parma in the lab of Dr. Maria Carreri. There he established methods to detect allergens and ailments, as food and food safety are critical aspects of the economy of his hometown. Next, he chose a PhD program at the University of Southern Denmark in Dr. Ulle Jensen's lab, where he developed methods for protein analysis. In 2014, he joined the laboratory of Dr. Benjamin Garcia at the Epigenetics Institute at the University of Pennsylvania for his postdoc. There, he optimized enrichment methods to link cell signaling cascades with chromatin changes. In 2019, he started his lab as assistant professor in the Department of Biochemistry at the Albert Einstein College of Medicine. So with that, I would like to thank Dr. Sidoli for being here and for running this workshop for us, and I will go ahead and pass it off to you.
Thank you. Thank you so much, and thank you so much for Protein Tech for the invitation. So this is by far the new record for number of attendees in one of my presentations. So now it's my responsibility to make this not boring. I'm going to start sharing my screen, and I trust that everything is okay. So I'm going to have a laser pointer here, and I'm going to start. So I'm going to present to you why we use immunoprecipitation-Mass spectrometry, how we use it, and especially I would like to spend most of my presentation trying to focus on how to troubleshoot problems, right? Because I don't want to use this presentation to go through a protocol. You know, there are many out there, you know, most of them work fine. The real challenge, especially for a scientist that is working at the bench, is to try to understand how you can fix things when something went wrong. So this is the agenda of my presentation. We're going to start with a brief introduction, how we prepare the sample. This is going to be the core of my presentation, like how to identify problems in your sample preparation and how to fix them. And then I'm going to tell you a little bit about data analysis. And for that, I'm going to make a little example based on the work that we do in our own lab. And I'll tell you how you can display and interpret results. So just a practical example of how IPMS is very useful for, for biological discovery. Okay, so very quickly, because this one is probably obvious for most of you who connected, you know, why do we study proteins, right? Because proteins, you know, especially compared to RNA, you know, we know RNA sequencing is more sensitive, is more reproducible, but we still like to study proteins, right? And the reason because we do is because proteins have a lot of unique functions that you cannot tell just looking at the sequence of an RNA, because proteins have different interaction, abundances, localization, modification. They can have different activities depending on their status. And obviously, interaction is one of the leading forces of why they have different activity in the cell. Interactions can be highly influenced, for example, by their modifications. We'll talk about that in a little bit. And modifications themselves, they can affect, you know, the stability of a protein. So think about ubiquitinylation, you know, how quickly they get degraded, if they are modified or not. Their localization, of course, and of course their activity. Like, so if there is a modification in the active site of an enzyme, of course the function of that enzyme will be different compared to if the modification is not there. So, you know, proteins and protein modification, they have a critical role in regulating the interaction of these proteins. So also, needless to say, the reason because we're so interested in proteins is not only for pure biological discovery, but also because they are almost always the major targets of drugs and therapies. So there are ways to study protein-protein interaction in the lab. Obviously, IPMS is not the only one. The other ones, I'm going to go through them very quickly. I'm not going to describe all of them, but some of them, they can quantify really the strength of binding, for example, between proteins or between a protein and a ligand. Examples are SPR, ITC. You need like pure proteins. They need to be very clean. There are, of course, approaches that they even allow you to tell exactly which atoms interact with which. I'm talking about crystallography, which gives very accurate information about protein interactions. There are methods like co-regulation. For example, you modify the abundance of protein A and you see how protein B gets co-regulated. These are more indirect effects, but they can still be used to tell the interaction between a couple of proteins. And then, of course, confirmation by co-occurrence. For example, you can take your two proteins that you think are interacting, you can centrifuge them, and then you can see if they precipitate together or if they separate together with size exclusion chromatography, for example. So there are many ways actually in the lab to check if two proteins interact with each other, and some of them give very high resolution level of information. But of course, the problem of all of these methods is that you need a hypothesis, right? You need to have your proteins already purified, and you actually need to know which one is the target. When we don't know that, that's when we use immunoprecipitation followed by some unbiased proteomic technique, of course, the gold standard being mass spectrometry. The protocol is really simple. There are some variations today. For example, you can cross-link proteins to conserve a bit more native interactions. You can, of course, label proteins in vivo. Now, turbo ID, bio ID is becoming very popular. And then, of course, you pull down these proteins with the streptavidin beads, because these are usually get labeled with biotin. You can, instead of using an antibody on a beads, you can use another protein. Now, in that case, we don't say immunoprecipitation because we're not using an antibody. We are saying pull down. So the protein immobilized on the surface could be another protein, could be a peptide, could be a modified peptide. We actually see a specific example in a minute. Or immobilized on some beads, on some surface, you can also use a small molecule as a bait. For example, it can be a drug, it can be a metabolite. There are certain experiments, for example, when you want to pull down kinases, the enzymes that catalyze phosphorylation, you can have beads decorated with the kinase inhibitor. And then what's going to happen is that the proteins kinases that interact with this inhibitor will be enriched in your pull down. The principle is very simple. So basically, you need to have beads. That the reason because you need beads is because you need to be able to separate them from the rest of the solution. You have your bait. When you do immunoprecipitation, is an antibody that is attached to the beads. We'll see in a second how. And of course, you need your input material, which is usually cell lysate that contains all the proteins of a cell. In some cases, it's a more simple mixture. And then, of course, whatever sticks to this antibody after the washes is going to be the target of your antibody. So when you choose what kind of consumables and reagents to use for this kind of experiment, there are certain steps that, to be honest, are not particularly critical. Like, so, cell lysase, we will discuss, is not very critical exactly to use very specific reagents. But for example, when you choose what kind of beads you want to have for your immunoprecipitation, it's important that you know what are the major types, right? So the material of the beads is usually either agarose or they are magnetic. And agarose beads, they are bigger. So they have actually a significant advantage because they are particularly large. They're up to 100 micrometer in diameter, and they have a lot of space, of course, to decorate with your antibody. And when you do your enrichment, you can actually use some kind of filters. For example, the one that you use to remove bacteria from your cell media, where you can basically block all of these beads on top of these filters, and then basically with a round of centrifugation, it makes it very quick, to wash them out after your bite. But because they are a little bit bigger and because they tend to be porous, they have a little bit more background. So you have to be careful about what controls to use, and we will discuss about that. Magnetic beads is an alternative. They are much smaller. They are easy to separate because you just use a magnet and then they get on the side of the tube. they are usually a little bit more expensive. So there are pros and cons. And of course, for these, because they are so tiny, usually you cannot separate them with filters. The coating of the beads, what is on the surface of these beads that allows you to, immobilize your bait, right? So if you're using an antibody, usually those beads are coated with protein A and protein G, which are basically proteins that they bind the constant region of the antibody. So this part right here. So this is actually very important because they actually not only you trap your antibody, but also your antibody is oriented in the right direction, to recognize your target, which of course is usually binding the variable region that is up here. Protein G, they usually have a little bit of a broader spectrum of which kind of antibodies they trap. Like protein A is a little bit more specific for IgG, but honestly, in most of the cases, the beads that you buy are usually coated with both proteins, protein A and protein G. So it's a mixture of both. Obviously, you need these if you want to do an immunoprecipitation. If you want to put down other things, for example, like biotin, then of course the beads are usually covered with streptavidin, or there are other kinds of beads that are coated with other kind of proteins. Like I said before, there are multiple types of pull downs. And Protein Tech, for example, offers a lot of these alternatives. Right? So there are also beads that recruit all of these proteins that are usually the focus of a lot of the major proteomic studies. Now, the antibody that is mixed with the beads, so then it gets attached and it gets properly oriented. Usually when you buy it, it's either polyclonal, which basically means that it's a mixture of multiple antibodies that they all recognize the same target, they all recognize the same protein, but not necessarily they recognize the same epitope. So they can interact with your protein in multiple directions. Honestly, for most of the work that we do, especially because we study proteins that are heavily modified, we tend to prefer polyclonal antibodies because proteins are present in different conformation with different modifications. And so to have a mixture of antibodies that recognizes multiple epitopes has its advantages, which of course, the disadvantage is that it's a little bit broader spectrum. It could create more background. Monoclonal instead basically means that you have a single protein, like it's just a single antibody sequence. Therefore, it's very specific because it targets one very specific epitope of your target. And that, has the advantage of being specific, of course, and the disadvantage is that if for any reason in that specific biological system, your epitope is masked by anything, your enrichment could fail. And then, of course, on top of the beads, we can, like I briefly mentioned before, we can have other types of baits. Like I said, we can have a peptide, we can have proteins, we can have other kinds of biomolecules. You can do enrichment of RNA interacting proteins by having a string of RNA on the beads, other type of biomolecules. And then, of course, as I briefly mentioned before, on the beads, they can also be a drug, for example. Okay, how the immunoprecipitation protocol works is essentially 4 steps. The first step is you have to prepare your input material. So you lyse cells and tissue, whatever you're using with the mild detergents. You don't want to use detergents that are too concentrated because then the problem is you will have your input material in very concentrated detergent, and this one will prevent the binding to the antibody when you actually mix the beads, the antibody, and your input material. Now, before you do the mix, the step number 2 is to bind the antibody to the beads. So the second step is first you take your beads, whatever material you decide to use, you take your bait, and then you incubate them a few hours. Usually we put them in a cold room or in a fridge at 4 degrees. We usually have them rotating, right? Because otherwise the beads, they all deposit to the bottom of the tube. And then, if you just wait a few hours like this, what happens is that they will bind to each other. And then, of course, the beads, they should be separated from the supernatant, washed a couple of times, because there will be probably antibody that is not bound to the bead, and this one will contribute to interfere with the immunoprecipitation. So you want to actually wash away the antibody that is unbound to the beads. Then step 3, at this point, you are actually ready to do your immunoprecipitation. So you have your beads coated with the antibody. Now you can add your input material, and your input material, same incubation, a few hours, spinning, keep them in cold, to minimize the activity of potential enzymes. And then what's going to happen is that you will have this product where your beads will be bound to the antibody, will be bound to your target. And then what will happen also that beside your actual target, there will be other proteins interacting with your target. These are the interactors of your target, which are also equally interesting. Actually, most of the time that you do an IPMS experiment, you know your protein is there. You want to know which proteins interact with my target. And then the final step, of course, after this enrichment has occurred, now we want to get the targets eluted from the antibody, so then we can analyze them by mass spectrometry, or today by other technologies as well. Usually, you can elute them in actually any format you want. You can use harsh detergents at this point. It doesn't matter. Your antibody will elute as well, but that's fine. It's going to be part of the mix. You can elute them in acid, which usually disrupts the interaction between antibody and targets. You can use a competitor. For example, you can put an excess of target, so then the antibody will actually release the proteins that are interacting to it. This one is a little bit dangerous, because if you add an excess of competitor, then this one it will start to interfere with your downstream analysis. Or, to be honest, my favorite, you can just wash the beads, and then you can add trypsin or whatever enzyme you're using for sample preparation for mass spectrometry, and digest the proteins directly onto the beads, which also acts as elution, right? Because you're going to destroy the antibody as well. You're going to destroy protein A and protein G. Everything will be in solution. And there will be, of course, interference from protein A, from the antibody, but that's fine. It's going to be separated by chromatography as you do mass spectrometry. And ideally, we want to put them in a workflow that looks like this. So basically, after we elute our proteins, we're going to digest them with trypsin, or we're going to digest them directly on the beads. In some specific scenario, you want to use enzymes that are not trypsin for mass spectrometry. We know that people who are connected, I'm sure they know that other type of enzymes that are used usually in special kind of applications, right? Because they tend to be a little less specific, a little less efficient than trypsin, that instead is designed to be, perfect for a regular proteomic analysis. And then once we have proteins digested into peptides, then of course they go through the proteomic workflow that is usually liquid chromatography coupled with MSMS, mass spectrometry. In an ideal scenario, everything worked out. At this point, you can actually display your results, and you can see, which proteins are enriched compared to a control. A control could be an antibody that doesn't bind to your, to your specific target. When another control could be the input material itself, to see actually what is just abundant in the cells, but does not specifically get enriched by the immunoprecipitation. Anyway, the point is that, as you know, proteomics is mostly based on relative quantification. So we don't, it's not just enough to say protein X, Y, and Z are detected in my sample, but you want them to be significantly enriched to the controls that you're using. And honestly, the more you have, the better it is. And then, like I said, when everything worked here, you have significantly enriched in your volcano plot proteins that are specifically interacting to your target. Ideally, here on top, there will be your target. Not necessarily, because it depends on how abundant your target is. Maybe it's very low abundance in the cell. So it may be that it's not the most enriched protein. That's fine. The important thing is that you enrich it with your immunoprecipitation. And then this is one way to display the results. We'll get to that a little bit later. Okay. Thing works, right? The point is, the reason I think you're interested in this presentation is because you want to find out what things did not work, right? So how do I identify problems in my experiment? Because yeah, it's okay if an experiment fails. If I don't know what went wrong, it's going to keep failing. So we're going to do in the next few slides, we're going to look at some results, some chromatograms, and then based on that, we are going to interpret what went wrong in my analysis. And here, these are some slides that I actually took them from a quiz that I usually do in some of my lectures. And then I ask students to look at these chromatograms and tell me what is wrong with them. Like, so this one is an example of a chromatogram where you had good results. And then here is an injection where you identified very few proteins and maybe you didn't even identify your targets, and you need to find out what went wrong in this specific experiment. Specifically in this one, now because this is not very interactive, I'm going to tell you the answer, but specifically what is happening is that you might have different kind of contamination. One of them is polymers. So polymers are usually plastic contaminants. They usually come from your buffer. They usually come from your consumables. They usually don't come from Eppendorf tubes, pipette tips, because these are made of polypropylene, that is a plastic that is it does not leak any polymer. But for example, in your cell lab, most of the material that you use is made with polystyrene plastic, and that one actually is prone to release polymeric compounds. So it could be that, for example, the plastic accumulated during the sample preparation in the cell lab didn't get properly washed. You can tell that these are polymers. You can tell it because you usually see in your chromatogram a train of signals that all have a kind of a regular variation in retention time, right? Because you're just adding one monomer, and then therefore the mass shift, the retention time shift is constant, and the mass shift is constant too, right? Because here, as you see, basically all of these peaks, they separate from each other from a plus 44 Dalton separation. Plus 44 Dalton, for example, could be a monomer that is CH2, CH2O, and then it repeats. And then you see actually addition of 44 Dalton. So this one is very clearly polymeric contamination. It has an hydrophobicity that is comparable to the hydrophobicity of peptide. So usually if you bring them with you up to the step of injection in the mass spec, usually they interfere with your final analysis. Usually it's not the only problem that you may have, but if it's significant, it can explain some of your poor results. Other example, you look at the spectrum, and then you see that instead of having your regular spectra with certain mass shifts, you see that most peaks, they have on their side a lot of other peaks with a lot of regular mass drifts. And this one is an example of salt adults. So if your sample has not been properly desalted, then you will see that most of the peaks will have this mass shifts corresponding to sodium shift or potassium shift. And this is a problem, right? Because now instead of having selected signals, now we have those signals that are diluted in many different conformations that are actually difficult to identify during the database search. Usually you can tell because a sodium shift has a mass addition of 22 Dalton and potassium 38 Dalton. And usually the problem comes if your peptides are not desalted properly, which you can either do it by doing stage tip desalting that usually is done before any proteomic experiment, or if you have a two-column system, so where the salt usually gets washed away by the trap column during the loading of your sample. If this doesn't happen and you have a lot of salt that ends up with you, basically sodium and potassium, they back exchange with hydrogen on the backbone of your peptides, and then you start to have those trains of signals that dilute the original signal and make your identifications much more complicated. Another problem is you simply don't have enough material and you basically have only trypsin in your sample. And that one you can tell if you look at your chromatogram, because there are a few tryptic peptides that even in the most fancy trypsin enzyme that you can buy from any catalog, there is a little bit of self-cleaving. So trypsin tends to cleave itself a little bit because, of course, it's a protease. And so there are a few peptides that are representative of trypsin cleaving itself. If those signals are actually more intense than everything else, it means that A, you use too much enzymes, and or B, the input material is much less than you thought it was. You know that usually we use trypsin on the fact of 1 to 10 to 1 to 100. So let's say 1 microgram of trypsin every 100 microgram of material to digest. So if you have results like this, it means that this was not even close to a ratio of 1 to 100. Probably used more trypsin than input material. Other example, your chromatogram looked very good when you run your standard, and now your peaks, they start to significantly tail. So basically come off very nicely in the beginning of the elution, but then they elute in a very asymmetric type of way. This one can be originated by two problems. One problem is overloading. So if you load too much on your column, then this means that chromatography will start to struggle, and that when you can tell because you underestimated actually the amount of material that you have. That overloading can also be caused by, for example, competition. Like I told you before, sometimes we elute our targets from our antibodies by using a competitive molecule. In this case, this was the flag tag peptide, and which we used to elute proteins that were flag tagged. And if we add too much of that peptide, that contributes to mess up your chromatography. That's why it starts tailing. That's why it peaks. They start overlapping with each other, and the mass spectrometer starts to struggle from having optimal results. Similar thing, tailing, you can have it if you have detergent contamination in your sample. This one is a problem that you solve at the stage of washing in your protocol, which was step 3 of the protocol that we discussed before. So you do the loading. The loading, you can do it in detergents. Actually, detergent, you probably used it to lyse your cells, you prepare your input material. After the binding occurs between your antibody and your targets, you need to wash the beads to eliminate everything that didn't stick to your antibody. That includes washing away very well detergent, especially if it's detergent like SDS, which is very sticky, and you need to do actually multiple washes. Actually, most protocol people use 4, 5, 6 washes to make sure that all the detergent has been removed. So then if afterwards, for example, you digest your proteins on beads, when you analyze your digest, you don't have detergent contamination, which again, you can see it because the quality of your chromatography significantly starts to suffer. And finally, another thing that, of course, is responsible for poor results. Another thing that could be caused by presence of detergent, but not only, is you see very little peptides in your run, and you cannot see what caused the problem. If you go towards the end of your chromatography and you start clicking down there in the spectra corresponding to the washing of your chromatographic column, basically at the very end of your chromatogram, you can see signals that look like this. So very highly dense charged molecules, which basically means that these are very large molecules, which basically means that these are big pieces of proteins that were not digested. So in other words, your trypsin digestion did not work efficiently, either because you didn't put enough or because there's something in your buffer that interferes with the trypsin digestion. A molecule could be EDTA, actually it does interfere with trypsin digestion. But the point is, if you go and click towards the end of your chromatogram and you look what kind of spectra are there, if you can find very large molecules that come off at the end of your chromatography, this is the problem. This is why you have poor results, is because you have you don't have good enough results. You don't have good enough digestion. Okay, additional problems that you cannot tell just by looking at the chromatogram, but we can tell in a few minutes when I'm gonna show you a little bit of examples for data analysis. Your enrichment failed. Maybe you did everything right, but for example, the antibody is not good enough. For example, your protein is not present in your input material, or the input material is not enough. You need to start from more, or the input material is contaminated by something else. For example, if you're growing cells and these cells, they have bacterial contamination, there's going to be a lot of proteins that are not part of your proteins of interest, and then they can interfere with proper results. And we will see some of these, how to diagnose when we are at the stage of data analysis, when we look at our protein table and what kind of proteins have been identified. You will notice that I didn't actually spend much time telling you how much material to start from and how much antibody to use. And the reason is because, of course, there are some standard starting places, but this actually may vary a lot depending on how good the antibody is, depending on how abundant your protein is in your cell lysate, depending on numerous factors. Right? So those are things that you cannot just put down on paper and it's always like this. So I would rather do a test run starting from reasonable amounts, and then based on the interpretation of the results, you can see how this can be optimized. Okay, so data analysis. For data analysis, I thought of the following. I'm going to spend like 3 minutes showing you an example of a project that we have in our lab. And I'm going to tell you how we solve our enigma by doing an enrichment with this kind of beads. And then we're going to move forward and see more in general how you approach data analysis when you are done with the NIPMS experiment. So just in 3 minutes, like I said, so in our lab, we study aging, right? So, and aging in particular, we are very interested in the structure of chromatin. When we study aging, chromatin, of course, being DNA and proteins that are, wrapped into DNA, specifically histone proteins that are heavily modified. As we get older, what happens is that our chromatin tends to be misfolded. So it basically loses its compaction state. The reasons are many, because as we get older, cells accumulate DNA mutation, because there are changes in epigenetics, because there are proteins that are contributing to keep the chromatin compacted that are no longer there. And then, of course, this happens, and then you have these domains of chromatin where there are strange combination of histone modification, transcription where you shouldn't have, and of course, in this region, they get recruited certain proteins that have functions that in young cells are not there. So the thing is, what we do in our lab, we basically study chromatin loses condensation, during aging. So we want to know if there are some molecules that actually protect chromatin that opens abnormally, and then basically, if there are these proteins, if there are these molecules, we can use them as therapeutics to basically delay the aging process. And the reason because I'm telling you this, because very quickly, we have a number of models in the lab where we actually have studied that some of these molecules exist. One of them is histone succinylation. I'm going to cut this very brief. Basically, if we use a progeny of centenarians, basically the children of people that have exceptional longevity, these are actually very good models to basically study what kind of molecular mechanism can protect against accelerated aging. And so what we see, like when we look at the chromatin of these children of centenarians, is that there is a certain histone modification called succinylation that is significantly enriched, while other markers that depict compact chromatin, instead they go down with time, which is exactly what we're expecting. So the point is, what is succinylation? Succinylation is a modification of histones that is not really extensively studied. Is similar to acetylation. Acetylation usually benchmarks open chromatin, but it's a little bit bigger and bulkier. It comes from succinyl-CoA, which is an intermediate of the TCA cycle. These two molecules are actually used exactly in the TCA cycle, and these are basically the precursors of protein succinylation, in particular histone succinylation that is relatively abundant. And so people that work with aging, they started to look at succinylation as a potentially organ protector. And there are some studies that they show that protein succinylation protect organs from damage, but how actually it works is still very unclear. So the interesting thing is that we know that succinate helps health span. Right, so if you actually give a succinate diet to mice for months, and these mice are old, actually they show improvement in health span metrics. For example, they have more grip, they are more good at balancing. They have actually, they have a number of features where they improve. So the point is, succinate and succinylation seems to be enriched in models of healthy aging. And interestingly, what we see is that if we actually feed cells with more succinate, we get more succinylation, and we get more histone modification that protect chromatin from decondensation. So this is a correlation, of course. Now the question is, how do you create an experiment where you show the causation that the more succinylation corresponds to more correspond to more silencing of the chromatin through these specific histone marks. And of course, we use it, we do it by immunoprecipitation or by pull down. In this specific example, what you can do, you can design an experiment where you take beads that are coated with streptavidin, and then you can produce or simply buy on numerous websites, you can buy peptides that are modified with your modification of interest. These peptides will bind to the beads, and then those are going to be your bait that you use to go fishing for proteins that interact specifically with this modification to identify the role of this modification in the cell. This one is an example of data that we have produced. You see that you can have an enrichment with the succinylated histone peptide, and then an acetylated histone peptides, you can use it as a control. And what you see here, you see a volcano plot where every dot is a protein of the nucleus of a certain cell line, which we use basically to tell which nuclear proteins interact with this modification or do not interact with this modification, because that is also very interesting. So, for example, if a certain histone modification prevents the binding of certain proteins, can also have a protective role. So always look at both directions when you look at your IPMS results, both the proteins that are being enriched by your favorite protein or modification, and look at the proteins that are being depleted, basically that do not enrich by your favorite protein. Now, in this specific example, all the proteins that are here on the left side of the volcano plot prefer protein acetylation, histone acetylation. And actually for the experts in the field, these proteins are very familiar because these are all proteins that indeed interact with protein acetylation. So they have a bromo domain. So a bromo domain is the domain that actually recognizes acetylated marks. And in fact, you see that they're all on the left side of the volcano plot. This was already a success for our specific experiments because you say, oh, basically succinylation prevents the binding of proteins with bromo domain. A lot of these proteins with bromo domain are transcription factors. Succinylation may reduce transcription from these regions where it's present. But the thing is, of course, if you look at the proteins that are enriched by succinylation, you can also find something. And now here specifically, we are zooming in on a specific class of enzymes. These are called lysine demethylases, basically enzymes that remove methylation from other protein. In particular, most of these, they act on histone proteins. So that's interesting. Succinylation recruits the methylases, prevents the binding of transcription factors. Long story short, we did a bunch of validation experiments where we actually showed that the active site of the lysine demethylases actually fits histone succinylation very well, and it probably actually contributes to the inhibition of this enzyme. And then, of course, there are some in vitro studies, enzymatic assays, where we show that indeed, if we use a succinylated peptide with some enzymes, there is an inhibition. And specifically, and this is the end of my story, the two enzymes that are inhibited the most by the presence of a succinylated peptide are the two enzymes that remove lysine 9 methylation and lysine 27 methylation. So basically means that if we have more succinylation, these enzymes are inhibited more by the presence of succinylation, which means that methylations on lysine 9 and lysine 27, the silencing marks, are naturally more abundant in the chromatin. So this is how we basically close the loop by doing an IP-MS experiment, or a pull-down MS experiment that actually, by identifying the readers of our favorite modifications, you actually can deconvolute the biology. So in other words, always look carefully about what kind of proteins you enrich, not only if your favorite suspect is there, but also what are the protein families that are being enriched in your IP-MS. And that brings me to the very final part of my presentation, just the last 4 slides, where I'm basically showing you some example of how to do interpretation of results. Again, when you do an IP-MS, usually you have your enrichment, and your enrichment is compared to a control, which again is typically background. It's maybe an enrichment against, again, an antibody that does not recruit your favorite protein. So what you end up with some proteins that are significantly enriched by your bait, and other proteins, of course, that are not. So the way to look at these results, basically the most interesting proteins in your table are the ones that are here on the top right of the volcano plot, because you have a very high fold change and you have a very high p-value, because in a volcano plot is expressed as negative logarithm. Of course, we know that p-value, the smaller the better. If you use the negative log, of course, becomes the bigger the better. So in other words, you want to know what is enriched on this side of the volcano plot compared to the opposite side. So open your Excel file with your results, with your identified proteins, and try to sort your table for the most interesting candidates. How do you do it? Basically, your most interesting candidate, they have a positive fold change, and they have a very high p-value. So if you create an extra column here, for example, column F, and then you multiply fold change and p-value, you get a number, sort that table by that number. On the top of the table, you're going to get the most, the proteins that are on the top right of your volcano plot. And this one already just visually could be helpful for you, because now you know that your interesting candidates are in the top of your table. But the fact that you sort the table the proper way, you can also use it for follow-up analysis with, for example, freely available tools. There are a number of tools that you can use from the internet. Of course, data analysis is very, open, depending on what kind of experiment you're doing. So I don't have time to go through every possible plot that you can generate, but I'm going to focus on the main one that usually are the basic pipeline of interpreting IP-MS data. As you know, most people start interpreting proteomic data from gene ontology enrichment. It's interesting. It tends to be a bit generic, the type of information that you get, but is a good first step. There are others, which we're going to see in a second, and these down here. First of all, gene ontology. Gene ontology, the reason because it's useful to sort your table from the most interesting protein to the least interesting, is because when you upload your gene list to a gene ontology search engine, some of these search tools, they actually calculate the enrichment of your terms based on which proteins are more at the top of your table. So use the right tools, for example, Gorilla, where you can upload a single ranked list of genes. And then with that, you can tell, basically, it gets weighted depending on how high in your list are your candidates. And then, of course, you get an enrichment, like I said, usually tend to be kind of generic terms. But it's useful because if you actually do an IP enrichment and you look at their gene ontology, and then you do the same thing with the background, and you sort of get the same terms, this one is very indicative that your enrichment did not work, right? Because you cannot have always the same term if you actually study the background, if you study your IP. That means that basically during your enrichment, you pull down mostly noise, which is the same kind of noise that you see in the background. So this one is helpful to, again, as a quality control. Then protein-protein interaction databases. As most of you probably know, String is a free tool. You just open it, you upload your list of genes, and then what it does, it basically connects your genes based on what is known from the literature in terms of how much these proteins interact. Interaction, keep in mind, they're not only physical interactions, some of them are genetic interaction, which basically means that they are known to be working together, but not necessarily touching each other in the cell. Still, it's very useful, right? Because if you actually upload your list into one of these tools, into String, for example, your bait should be there, right? And then you probably should find some interactors. If this protein has been studied before, you might want to find them to be, you might want to see your protein not completely isolated in the network, but connected with some proteins that are known to be interacting with it, and they are present in your list. And then if you have targets that, for example, are part of a complex, it tends to be reassuring if multiple proteins of the complex are in there. So in other words, it's for example, if you pull down the PRC2 complex, then you expect to see the protein EZH2, SUDS12, EED. You expect to see all the proteins being enriched in your IP, and it's easy to see because they form subnetworks in the String database. It's not the only database String that you can use. There are some others. To be honest, the others tend to be a little bit less annotated, but some of them are really useful because, for example, are manually curated, and String unfortunately is not because it contains too much information. But for example, Bioplex is a nice database because actually protein-protein interactions that have been obtained using mass spectrometry data. So it's actually a nice validation with the experiment that you do because it's probably also based on mass spectrometry. Very quickly, the last two, protein domain analysis. This one is very helpful, for example, if your bait, for example, is a modification or your bait is a specific motif. Now, for example, like I said before, acetylation. So we know that acetylation brings down proteins that contain bromodomains. So you have your list of candidates. You can paste your list of candidates on UniProt. You go to retrieve, you retrieve all the information that you want about the proteins that you have identified. And you can check, for example, if among the proteins that you enrich, how many of them contain bromodomain. This is also a nice validation to basically confirm that what you're pulling down actually makes sense biologically based on what we know, not on the function of the protein, but on specific domains of these proteins. That there are specific regions of the proteins that you pull down that make sense, that are enriched by your bait. And that's why this one you can do it any kind of proteomic experiment. And finally, exploit the capabilities that we have from public resources. Like, so by now, there are thousands and thousands of proteomic experiments that are available online, and there are some tools that basically study covariation. Or, in other words, basically what it means is that you have these hundreds of data sets, of thousands of data sets, where proteins actually regulate together. Like, so in study one, so the protein is very abundant, and so is protein B. In other study, the protein is low abundant, and so is protein B. And then by doing this, what you do, you create some kind of associations of proteins that are always regulated together, and therefore they probably interact in some sort of form. And so you can actually upload the list of your targets that you have identified on these databases. This is an example of two software that can do the job. And then you can check, are these proteins known to co-regulate? Are these proteins known to be always, with similar regulated abundance among this plethora of data sets that we have available online? That one actually is very useful to confirm that probably your proteins, they have some biological interaction and is worth investigating further, or at least, prioritizing some of the candidates. With that, I'm done, actually right on time. And I'm going to conclude basically by presenting again the first slide with the agenda. I hope that it was useful. I hope that we spent a good amount of time discussing why IPMS is important and what are reagents and consumables that are worth considering during your prep. I wanted to spend most of the time of the presentation discussing potential problems and how to identify those problems. So then your trial number 2 has a much higher chance of being successful. And then, the final part of the presentation, a practical example of how you interpret IPMS results and, why there's actually a logic also in the steps of the data analysis in order to interpret the results of your experiment. And this is actually really my final slide, picture of the lab, funding, a lot of people that work with us. And of course, thank you for Protein Tech for the invitation. And now I guess it's question time.
Wow, what a wonderful presentation, Simone. I mean, you can look at the chat. Everyone is just very grateful for your kind of a laid out presentation. I mean, for me, it was really valuable that you went through the typical problems and characterizing the different peaks, because mass spec data is always very difficult to interpret. So that was very useful for me. We have a lot of questions to get through, so we're not going to get to all of them, but I do want to make sure I picked out a few. But before we do that, I do just want to share a few more slides. I'll go through these really quickly, and then we will begin the Q&A. Okay. So before we begin the Q&A, I just want to spend a couple minutes introducing our nanobody-based reagents for IP. So as Simone mentioned in his talk, so nanobodies can be used for the efficient and clean pull down of target proteins. So nanobodies, or VHHs, are actually the antigen binding domains from heavy chain only antibodies that are derived from alpacas. And they're quite small, so they're only about 15 kilodaltons. So they're actually about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity. So at Protein Tech, what we've done is we've actually created a portfolio of products that are centered around nanobodies, including our Nano-Traps, which are nanobodies that are conjugated to agarose beads for IP. So I'm going to talk a little bit more about the Nano-Traps. So they do offer significant advantages for IP, especially in IPMS experiments where sample purity can really make or break your experiment. So because a nanobody is simply the binding domain of a heavy chain antibody, it actually doesn't introduce heavy or light antibody chains into your samples. So that results in much cleaner data. Also, because the nanobody is actually covalently attached to the beads, it stays bound to the beads even after elution, which further helps with your sample purity. The Nano-Trap also, they have high affinity for their target protein, which allows for short incubation times while still being able to achieve highly efficient pull downs, even for proteins that are expressed at low levels. And then another key benefit is their stability under stringent buffer conditions. So nanotraps can actually tolerate wash buffers that are containing things like reducing agents, chaotropic reagents, high salt, or harsh detergents, and they're also fully compatible with on-beat digestion. So altogether, these features make nanotraps an ideal tool for helping you generate, reliable, high-quality data for your IPMS experiments. So I really quickly just want to share one piece of data as an example of the quality IPs that can be generated by the nanotraps. So here we're comparing how our GFP trap performs to a conventional GFP antibody. So we're showing here two SDS pages, and we're showing our input of the lysate, the flow through after our IP, and then the bound fractions for each of the IP. I'm just going to get the laser pointer out here. And you can see that in the conventional GFP antibody gel, it shows the band for your GFP that corresponds to GFP, but you also see these additional bands, and that's going to correspond to your heavy and light chains of your GFP antibodies. And you don't see that in the SDS page from the trap. So you can immediately see that the trap is allowing for a cleaner pull down. Then if you look at the blots here on the bottom, you can see the efficiency of the pull downs. So when using the Nano-Trap, there is no GFP protein that's left in the flow through, and it's all enriched here in the bound fraction, which shows the effectiveness of the pull down. And this is because the traps actually have incredibly high affinity that goes up to even 1 picomolar, which allows it to capture all of the protein of interest. However, with the antibody, you can see that there is enrichment in that bound section, but there is a small amount of that protein that is remaining within the flow through, meaning that you have a less effective pull down. So in cases where you're working with applications downstream of IP that require high purity and pull down, Nano-Traps do offer these important advantages for you. So this is our current Nano-Trap offering. Most of our Nano-Traps are against tags. However, we do have a few traps that are available for oncology targets. So our tags that we have are things like fluorescent tags, peptide, and solubilization tags as well. Our nanotraps are also available in various formats, including the nanobody that's conjugated to agarose, magnetic agarose, and magnetic particles M270. And if you'd like to try a nanotrap, you can request a free sample as an attendee of today's webinar by scanning the QR code and filling out the form. I'll have this information on the last slide too, and we'll also email you the form as well. I also just wanted to introduce to you all a new product that we've launched. So these are our anti-IgG VHH beads for IP. So these are going to be our ready-to-use VHHs that target rabbit and mouse IgGs and are coupled to agarose beads. So these are actually designed to be a better alternative to using protein A and G beads, and they bind to the antibody that you're using for your pull downs. So this product provides a solution for the headaches that come with using protein A and G beads, such as unspecific FC binding or the leaching from beads that can compromise specificity and increase your background. So this product was actually optimized for targeted precision to ensure cleaner results and has redefined specificity and purity in IPs. So if you do want to learn more, you can scan this QR code here. You can also go onto that page and request a free sample of the beads as well. And then lastly, I just want to mention that as an attendee of today's webinar, you will receive a couple follow-up emails. The first will include a recording of this webinar, as well as links to the 2 request forms that I just told you guys about right now. So the free sample for the Chromotech Nano-Trap, as well as a free sample for an anti-IgG VHH bead. And then you will also receive another email that will have a certificate of attendance for today's webinar. So with that, I think we can go ahead and begin the Q&A session. If you do have any questions after the Q&A session is over, you can always email us at events@ptglab.com, or you can reach out to Simone directly. I put his email right here. And then if you did like today's webinar and you want to learn more about our upcoming events, please do follow us on social media because we post about all our upcoming webinars on there. All right. So there is quite a few questions. I've selected a few. The first thing that I noticed, there was quite a few questions about asking, how can you confirm that you've had a good elution?
Sorry, I was actually muted and answering some of the questions live. So you can see in the Q&A tab that some of them have been answered. Really good question, all of them. Sorry, I was distracted by that. Can you quickly repeat the question?
Yeah, no problem. There were a few questions in the chat that were asking, how can you confirm that you've had a good elution?
Yeah, good question, as all of them. The thing is, if you use some of the practice that I listed in my presentation, like digestion beads, you cannot really quantitate, how much elution you got. But if the digestion is efficient, of course, you will see that peptides are all being digested properly in your chromatogram, and you will see very little intact proteins left in your chromatography. So that's the way I would use to calculate the efficiency of elution, right? Because the point is, yeah, you can quantify your proteins after you do the elution, but to be honest, if you don't know in advance how much of your protein is present in the cell lysate and how much targets it pulls down, that quantification is kind of meaningless. So I would actually use the efficiency of digestion as measurement of elution efficiency.
Great, thank you. The next question is asking, how do you troubleshoot cross-linking the antibody to beads to prevent eluting the antibody?
Yeah, that's a good question too. Some people, after they conjugate the antibody with the beads, they use cross-linking to make sure that it doesn't detach anymore. To be honest, if your input material is in a buffer that is reasonably mild, it should not detach. And if it does detach, there's probably also a good chance that you will lose the interaction between the antibody and your targets. So in my personal opinion, maybe some people would disagree, I would say cross-linking is probably not necessary, because one of the problems with adding cross-linking is that you actually risk to modify the variable region of the antibody where the interaction of your target should occur. So my personal preference is to not cross-link the antibody with the beads. However, if you do it, there's nothing wrong with that. Just keep in mind that the cross-linking tends to be a little bit stochastic. So it doesn't necessarily mean that you will have 100% efficiency. So you can still find antibody in your elution. You can still find antibody in your mass spec runs. I think, sorry, I spend an extra second on this question. I think the reason because this question is being asked is because people tend to be worried that the bait, so your antibody, ends up suppressing the signal of your results in your chromatography. What I would do, to be honest, instead of adding chemical, addition to your experiment, I would probably solve the problem by expanding the chromatography in your LC-MS run. You have better chance of detecting more this way than by chemically cross-linking the antibody to the beads.
Great, thank you. And kind of a related question that had come up was, do you have any suggestions on how you can recover the capturing antibody from the elute?
Recovering. No, actually, that's a tricky question. I don't know. I mean, of course, you can add protein, beads with protein A and protein G to the mix, but you are going to recover not only your antibody, you are going to recover all antibodies that are present in that lysate. So I don't have a good answer. I don't know if you have a good answer, like experts at protein tech, if they were
you're the expert here.
Yeah, sorry, that one is a question I don't know how to answer. Yeah.
If you do want to email that question over to the events email, we can reach out to our R&D team and see if someone has an answer for you. So the next question is asking, how do we clarify if the interaction between the protein that's immunoprecipitated and its target is direct or indirect?
Yeah, exactly. Yeah, something I haven't touched enough on my presentation is the fact that when you do this immunoprecipitation experiment, you pull down a network of interactions. You don't have the exact information about physical touch between proteins. The way to answer this is either you do a cross-linking MS experiment where you cross-link targets with each other, and then you can identify proteins cross-linked to each other. That's one good direct way to identify direct interactions. Otherwise, in my personal perspective, but I come from a biochemistry department, so other people might disagree, I think the best experiments to do are the ones that I introduced in the very beginning of my presentation. You have your hypothesis on your bait and your target. You can buy these proteins, you can purify them. Most universities, they have a protein production facility that they usually purify those proteins at not very high costs. And then you do one of these experiments like surface plasmon resonance or titration calorimetry, and then basically you get not only direct interaction, but you get the strength of the binding. And that one really goes a long way for high impact publications.
Great, thanks. The next question is, what is the advantage, if there is one, to bind the antibody to the beads before to the lysate compared to incubating the antibodies with the lysate and then adding the beads?
Yeah, so I don't have direct data of comparing the two approaches, to tell you exactly which one works better, but I can tell you that the vast majority of laboratories that do this kind of experiments, first they incubate the beads with the antibody, and then they incubate the lysate. And the reason because you do that is because if you first incubate the antibody with the lysate, it becomes very hard to wash away antibodies that is unbound to your target. And so basically, you're going to pull down a lot of antibodies that are bound to background or antibodies that are bound to something else. So you have the experiment a little bit less under your control.
And then there's another question asking, what are your thoughts on depletion of high abundant proteins? Is it beneficial? And if so, are there any kits that you recommend?
It's very beneficial in the sense that is honestly the best way in mass spectrometry to increase your sensitivity, to fractionate your sample. In my experience, kits that just remove selected proteins, they have limited efficacy. Maybe it's just me that I don't know how to use them. One thing that works well for me is to actually fractionate your input material with different kind of orthogonal chromatography. For example, size exclusion. So you can actually split your input material instead of loading all of it on the beads, split it into 10 fractions, either by size exclusion or helic chromatography or C4 chromatography, and then do an incubation for every fraction. That obviously multiplies the number of injections that you have to do in the mass spectrometer afterwards, but that one is by far the best way to improve sensitivity in any kind of proteomic experiment, not just IPMS.
Great. So there were a few questions, so I'm going to combine these questions that were asking about what types of acids you can use for elution, and then how do you then neutralize the elute?
Yeah, we have used multiple ones. We have not noticed any significant difference between them. So the most common one is acetic acid. You can use formic acid. You can use trifluoroacetic acid. To be honest, it doesn't really matter too much because normally, and if you don't do it, I recommend doing it. The step that comes, after the elution exactly is digestion, right? And so you have to use a significant amount of buffer to neutralize the acid. So whichever acid you use, you still need to use these buffers regardless. Related to this though, I would recommend once you have achieved the low pH that you need, I would recommend to not exaggerate with the amount of acid that you use. That is not going to help more the elution, or even if it does, it's going to significantly interfere with your digestion. So at the end, results are worse.
Okay, that's good to know. And then I think we can end with one more question. I know we're a little over, but I wanted to make sure we got through some of these. This one just came in. What are the pros versus the cons of on-bead digestion versus urea elution?
Yeah, I mean, I say like the on-beads digestion is, like I said in my presentation, my favorite because first of all, it's simpler. It combines digestion with elution. You are basically almost 100% sure that all of your proteins fall off from the beads because you're basically chopping off also the antibody and the protein that coats the beads. Eluting with other buffers, for example, like urea, they may have the advantage of retaining some of your antibody on the beads, so that interferes a little bit less to the final analysis. that's fine. But again, you cannot use conditions that are too harsh, because then you're going to have a problem at the downstream digestion step. So the advantage of using urea is probably not because it's better to elute. It's probably because it contributes to unfold your proteins. And then, if you use it below 2 molar, it actually improves the efficiency of digestion. It does not necessarily make the elution better, but it may improve the efficiency of digestion.
Great. All right. Well, thank you so much, Simone, and everyone who attended. I'm just going to pop this last slide back up so that you guys have the contact information. So again, if you guys have any questions, you can always email us at events@ptglab.com. You can also reach out directly to Simone too. I put his email right here. And with that, I think we can go ahead and end the webinar. I'm sorry if we didn't get to your question live, but you can always email us if you want. And with that, I will go ahead and end. And I hope everyone has a great rest of your morning, afternoon, or evening, depending on where you're tuning in from the world. So take care.
Thank you for connecting.